learning outcome number 3 for Module 4, we're going to be covering the bone histology. Just like any other tissue, bone tissue will contain several cells that are going to be surrounded by an extracellular matrix. The cells in adult bone, they are for most part osteocytes that will be surrounded by this extracellular matrix that is formed by water, collagen fibers, and crystallized mineral salts. The mineral salts, they're what gives our bone the hardness consistency, and the collagen fibers gives the bone some flexibility, which will provide the tensile strength and the resistance to being stretched or torn apart so that they don't break easily. In summary, we can say that although a bone's hardness depends on the crystallized inorganic mineral salts, the bone's flexibility will depend on its collagen fibers. In fact, to test this out, if we soak a bone in an acid solution such as vinegar, this will dissolve its mineral salts and it will cause the bone to become very rubbery and flexible. With regards to the composition of the extracellular matrix, I know we talked about what's going to be in them, but more specifically, it will contain 15% of water, it will contain 30% of collagen fibers, and the rest, which is 55%, will be of these crystallized mineral salts. Most of the mineral salts are going to be calcium phosphate, which will combine with calcium hydroxide to form these crystals that are then called hydroxyapatite. Then the hydroxyapatite crystals will combine with other minerals like calcium carbonate, magnesium, fluoride, potassium, and sulfate, and all of these will form a big crystal-like structure in between the collagen fibers that will then harden and surround the osteocytes. This process of crystal-like deposition and hardening is called calcification. There are 4 main cell types in bone tissue, and they are listed here. There are the osteoprogenitor cells, which can also be called osteogenic cells, the osteoblasts, the osteocytes, and the osteoclasts. 3 of these cells will come from a bone cell lineage, which are the osteogenic or osteoprogenitor cells, the osteoblasts, and the osteocytes. Basically, one will give rise to the other, starting from the osteoprogenitor cells, which are really stem cells that originate from the mesenchyme cells. The 4th cell, which are the osteoclasts, they actually originate from the white blood cell lineage. Let's talk in more details about each of these cells in the next few slides. Osteoprogenitor cells are these unspecialized bone stem cells that are derived from mesenchyme, and it's the tissue from which almost all connective tissues are formed. Just so that you know, genic means producing, and now that you know this definition, it might make it easier for you to understand the function of these cells. As they are the only bone cells to undergo cell division, the resulting cells from this division will develop into osteoblasts. Osteoprogenitor cells are found along the inner portion of the periosteum, in the endosteum, and in the canals within bone that contain blood vessels. The periosteum is the lining of the connective tissue around the outer portion of the bone, as we can see over here. Just like we talked earlier when we covered cartilage, remember that we had a structure of connective tissue called the perichondrium that covered the outside of the cartilage. Well, it's the same concept with a similar name, but now instead of it being named after cartilage, it is named after a bone. The endosteum will cover the bone on the inside, therefore it will line that cavity within the bone, as we can see over here, which is called the medullary cavity. And the canals are these tunnels within the bone itself where blood vessels will be present, and we will see the canals when we cover the histology of the bone. Osteoblasts are bone-building cells. They will actually be the ones responsible for building the extracellular matrix. Therefore, they will synthesize and secrete collagen fibers and also other organic components that are needed to build this extracellular matrix of the bone tissue. And if they are the ones that are responsible for depositing the matrix, they will be the ones that are responsible for the initiation of calcification. As we have discussed already, the calcification is going to be the deposition, the crystallization, and the eventual hardening of mineral salts in the framework that is formed by collagen fibers of the extracellular matrix. Since this extracellular matrix will be hardening, the osteoblasts will eventually surround themselves with this hard extracellular matrix. Therefore, they will become trapped in their own secretions, and eventually they will mature into osteocytes. It is also important to note that cells that end in blast in bone or any other connective tissue secrete extracellular matrix. Also, another tip to remember the functions of the osteoblasts, as they are similar in name to the osteoclasts, and this can be a little bit confusing, is that osteoblast has the letter B, therefore they will be responsible for building the bone, as they are the ones responsible for producing the material responsible for the formation of the matrix. Again, B for building. Or if you prefer to define the term, blast means buds or sprout, like producing something new. For me, it doesn't really matter what you use. You can even create your own little rules to remember the different types of cells, whatever is better for you to remember. Moving on to osteocytes, like I mentioned on the previous slide, osteocytes will be the mature bone cells that derived from the trapped osteoblasts within the matrix. They are the main cells in bone tissue, and they will maintain their daily metabolism, such as the exchange of nutrients and wastes with the blood. Like osteoblasts, osteocytes do not undergo cell division. As you remember, like we said earlier, the only cell type that do undergo cell division are the osteoprogenitor cells. Also, if you recall, cells that end in cyte, in bone or any other tissue, will maintain and monitor the tissue. Now we're going to cover osteoclasts, which are the last cell type present in bone tissue, and they are actually very different from the other cells we mentioned earlier, not only in terms of morphology, but also in terms of its functions. Starting with how big these cells are compared to the other cells, and the fact that they have several nuclei, as you can see here on this image. The reason why they are so big and contain several nuclei is because they are formed from the fusion of as many as 50 monocytes. Monocytes are a type of white blood cell that also give rise to macrophages. These osteoclasts will be concentrated in the endosteum, which is the inner lining of the medullary cavity. As you can see, there is an area of the plasma membrane of the osteoclast that has several deeply folded invaginations right over here, forming more of what we called a ruffled border. This ruffled membrane will be located on the side of the cell that faces the bone surface, and this will increase its surface area, just like the microvilli. The reason why they face the bone is because these cells will be releasing these powerful lysosomal enzymes and even acids that will digest the protein and mineral components of the underlying extracellular matrix of the bone. This breakdown of extracellular matrix of bone is what we call bone resorption. Cells that end in clast are responsible for bone breakdown in the extracellular matrix. The way that I remember the function of osteoclasts is that they contain the letter C, and C is for chewing, or the meaning of the word clast means breakdown. Therefore, the osteoclasts are responsible for chewing up or breaking down the bone in response to environmental, normal, or abnormal cues. And like I mentioned earlier, this process is called bone resorption. Therefore, resorption is part of the normal bone development, the growth, maintenance, and also the repair of bone. Osteoclasts, they help to regulate bone calcium level in response to certain hormones. We will talk about this regulation when we cover calcium homeostasis on our last learning outcome of this module. These cells are also the target cells for drug therapy used to treat osteoporosis. As you can imagine, these cells are the ones responsible for the excess chewing of the bone in individuals that present with this disease, which is a weakening of the bone tissue. As bone formation progresses, the newly formed bone goes through several stages. Remember that ossification, which is the formation of new bone, can occur several times during an individual's life, like starting with the fetus stage, when increasing in length also, and any time there needs to be a repair of a fracture, for example. There are two types of bone formed during ossification. We have what we call the woven and the lamellar bone. The woven is the first type of bone that osteoblasts form during ossification. It is fairly weak because the collagen fibers are going to be randomly oriented in many directions. After woven bone formation, the osteoclasts break down the woven bone, and the osteoblasts will build the stronger, more permanent lamellar bone. Therefore, we can say that the lamellar bone is what we call the mature bone. This bone will be organized into this thin concentric sheet or layers that is called a lamella. That's why it's called the lamellar bone. As this ossification process continues, bone is going to be further modeled and shaped into its final form. Whether bone is of the woven, weak type or the lamellar, more mature type, it can be further classified into spongy or compact bone. And this second classification takes place according to the amount of bone matrix relative to the amount of space within the bone. We will focus more on spongy and compact bones instead of woven and lamellar bones on the next couple of slides. There are two main types of bone, the compact bone as seen here and the spongy bone, which we will cover in the next slide. Compact bone can also be called cortical or dense bone, and this is the bone that will line the surface of all bone types. In long bones, it will be mainly present in the diaphysis, which is the center part of the bone, as we can see right over here. When you look at a bone, you're looking at the compact bone specifically, and it looks pretty dense and solid. However, when you analyze it microscopically, as we see over here, you notice how the compact bone is quite porous and it will contain several spaces or tunnels or canals. The function of compact bone is to provide the protection, the support, and also to resist the stresses that are produced by weight and movement. The functional unit of a compact bone is what we call an osteon, as seen right over here, or also called our Harvasian system. Each osteon is going to consist of these concentric lamellae, as seen right over here, that are arranged around a central canal, which can also be called Harvasian or osteonic canal, as we can see on this image. These concentric lamellae that sort of resembles the growth rings of a tree, they are circular plates of mineralized extracellular matrix of increasing diameter. This just means that the circles that are on the inside are going to be smaller than the circles that are on the outside. As you can see, they will surround the central canal, and inside the central canal we find a small network of blood vessels, arteries and veins, nerves, which are represented here by the yellow, and also lymphatic vessels, which are represented here by the green. Between each of these concentric lamellae are these small spaces that are called lacunae, and we can see the lacunae better on this other image over here to the right. Lacunae means little lakes, and these little lakes are going to contain the osteocytes that, remember, they originated from the osteoblasts. This image over here on the top right shows you how the osteocyte looks with all these projections coming from the cell body. The cell body is what's going to be located in the lacunae. Because the osteocytes will have several projections, these projections will form even smaller canals, that are called canaliculi. Canaliculi means small canals or small channels. And these canaliculi, they're going to be filled with extracellular fluid that are coming from the osteocytes themselves. Now, these neighboring osteocytes, they will communicate with each other via a gap junction. Therefore, the canaliculi, which again contains the cell projections, they will connect with the lacunae, which is that area where the cell body is located, with one another and with the central canals, forming this very intricate miniature system of interconnected canals throughout the bone. The advantage of this communication is that it provides many routes, as you can imagine, for the nutrients, the oxygen, to reach the osteocyte from the central canal and for the removal of the waste products. The interesting thing about osteons in compact bone tissue is that they are aligned in the same direction and they are parallel to the length of the diaphysis, which again is the center of the bone. As a result, the diaphysis of a long bone is actually able to resist the bending or fracturing even when considerable force is applied from either end. Also, compact bone tissue tends to be thickest in those parts of a bone where the stresses are applied in relatively few directions. These lines of stress in a bone are not static. This means that they change as a person learns to walk, or for example, in response to a repeated strenuous physical activity, such as weight training. The lines of stress in a bone can also change because of fractures or even physical deformity. Therefore, the organization of the osteons is not static, but it changes over time in response to these physical demands that are placed on the skeleton. These areas between the neighboring osteons, they will contain a lamellae that's called interstitial lamellae, which also have lacunae with osteocytes and canaliculi. This interstitial lamellae is going to represent the fragments of older osteons that have been partially destroyed during bone rebuilding or during growth. The blood vessels and the nerves from the periosteum, which is this membrane over here of connective tissue that surrounds the bone, will penetrate the compact bone through the perforating canals, also called interosteonic canals. The blood vessels and nerves of this interosteonic canal will connect with those of the medullary cavity, the periosteum, and the central canals as well, as you can see from these images. Arranged around the entire outer and inner circumference of the diaphysis of a long bone are these structures that are called circumferential lamellae. The circumferential lamellae directly deep to the periosteum are called external circumferential lamellae. They are connected to the periosteum by these perforating fibers, and the circumferential lamellae that line the medullary cavity are called internal circumferential lamellae. In contrast to compact bone tissue, spongy bone tissue, which is also referred to as trabecular or cancellous bone tissue, does not contain osteons. Spongy bone tissue will always be located in the interior of a bone, and it will be protected by a covering of compact bone. Similar to compact bone, it consists of lamellae that are arranged in an irregular pattern when compared to compact bone, as we can see from these images. This lamellae is made up of thin columns that are called trabeculae, where trabeculae means little beams, and that's where the name comes for trabecular bone. Between the trabeculae are these macroscopic spaces that are filled with either red bone marrow in bones that produce blood cells or yellow bone marrow in bones that produce adipose tissue. Both types of bone marrow contain numerous small blood vessels that will provide nourishment to the osteocytes. Each trabecula will consist of these concentric lamellae, as you can see from the image, in addition to osteocytes that lie in the spaces called lacunae, and they will also have the canaliculi, as seen on the image, that radiate outward from the lacunae and allow communication between the osteocytes. Spongy bone tissue makes up most of the interior bone tissue of several types of bones, which are short, flat, sesamoid, and irregular shaped bones, and these bone types will be covered in the next learning outcome. In long bones, it forms the core of the epiphysis, which is the ends of the bones as seen on this image, and it will be surrounded by this thin compact bone for protection. In the diaphysis, it will form the narrow rim that will border the medullary cavity. At a glance, the trabeculae of spongy bone tissue may appear to be less organized than the osteons of compact bone tissue. However, they are actually precisely oriented along lines of bone stress, which is a characteristic that helps bones resist stresses and transfer force without breaking. In fact, you might think that the compact bone is stronger than the spongy bone, which to a certain extent might be true. However, the arrangement of the trabeculae make the spongy bone resist these stresses from several directions, making it a tough bone as well. Therefore, we can summarize that spongy bone tissue is different from compact bone in two respects. First, spongy bone tissue is light, which reduces the overall weight of a bone. This reduction in weight allows the bone to move more readily when pulled by a skeletal muscle, making it easier for us to move. Second, the trabeculae of spongy bone tissue support and protect the red bone marrow. Spongy bone in the hip bones, in the ribs, in the sternum, vertebrae, and in the proximal ends of the humerus and femur is the only site where red bone marrow is stored, and therefore is the site where hemopoiesis, which is blood cell production, occurs in adults.